Hi, I am Sriram Krishnamurthy from Chemical Engineering Department at IIT Madras. The amyloid beta protein belongs to a class of proteins which self-assemble into long thread-like aggregates. These aggregates are called fibrils. The deposition of such insoluble fibrils on the neuron cells are considered as the major cause for the Alzheimer's disease. If fibrils cluster together to form insoluble plaques and deposit on the cell membrane, these plaques inhibit the neuron cell communication and ultimately lead to cell death. Apart from their implications in various diseases, amyloid forming protein have applications in various emerging areas of research owing to the structural stability and biocompatibility. One major example is the development of biosensors using functionalized amyloids. Recently, there has also been interest in using these fibril forming proteins for tissue engineering research. It has been known for quite some time that the amyloid beta protein tends to aggregate at a faster rate when it is under flow. Studies with different amyloid forming proteins such as amyloid beta, insulin, and lactoglobulin, etc., confirm this. And the general consensus is that the shear flow accelerates the formation of fibrils. But there is a lack of understanding about the problem in terms of the microscopic picture. My research aims to throw some light into the underlying mechanism at multiple length and time scales. Such an understanding is not only important from a diagnostic perspective, but also from the industrial standpoint, where proteins like insulin are subject to a variety of shear forces during its manufacture. With that background, I designed the experiments with my colleague to understand the kinetics of fibrillation of A beta protein. We shared the protein solution in two different flow environments, one in a controlled manner inside a quid cell and another inside an orbital shaker with haphazard flow. We monitored the concentration of fibrils formed over time. Normally, these proteins would take months to form fibrils under undisturbed conditions, but in both cases, we observed that the whole reaction takes place orders of magnitude faster. But the behavior is starkly different from both the condition, as is visible from the plots. The most important difference is the existence of different decreasing fibril concentrations for decreasing shear rate in the controlled shear environment. To explain this, I developed a population balance model aided from previous literature. The protein monomers form a nucleus and then proceed to elongate into linear aggregates. The aggregates could also break into smaller fragments and continue to grow. The novelty in this work is the consideration of depolymerization step. In this step, we hypothesize that the monomer in the fibril can actually erode away depending on the applied shear rate. To our surprise, we found that the effect of increasing the shear rate is to actually reduce the erosion of monomers from the fibrils. Now, confirming this mechanism experimentally would require setting up of complex experiments, which can be time consuming. Therefore, we use molecular dynamic simulation to perform computer experiments and understand this finer detail. Towards the end, we developed a coarse-grained model which can replicate the aggregation behavior of these proteins under shear. The model consists of spherical beads with attractive patches. These patches mimic the attractive hydrophobic interaction between the protein that is considered to be crucial for their aggregation. From the simulation, we get linear aggregates reminiscent of the protein fibers. This structure is also similar to cylindrical micelles formed by surfactants at favorable conditions. Performing shear simulations on such aggregates will give us a better understanding of the problem. Thanks.